looks like the big hand is on the 2, the little hand is on the 7, so it must be 10 o'clock. So, Jerry, if you pass the bleach, we can get started.
Okay. All right. Well, today I'm going to give a COVID update, but I thought I would just start with a little backdate, have a little fun with what has gone on with the COVID virus, with all the mania in the news.
I thought you were going to do a Yiddish lesson.
I am. That's part of it. Yiddish has some very important expressions that aren't contained in other languages. And one of them is a gonif, which is Yiddish for one who transgresses moral or civil law, a dishonest person. He's a gonif. Okay. So hydroxychloroquine and chloroquine were subject to a lot of hysteria on both sides. It's always fun to watch laymen talk about science. And what we had was, we had a huckster on one side, and we had the media mania, Trump derangement syndrome on the other, and it was totally non-scientific. And it was just hysteria, but they're in the job of ratings, not in the job of science. And they make it their business to be ignorant. So I thought I would talk just a couple of minutes about the craziness that went on with hydroxychloroquine. Now, first, hydroxychloroquine and chloroquine have biological effects, and people have been on chloroquine for many years for the prevention of malaria. Now, the way the hydroxychloroquine, to show you it wasn't a crazy idea, the way hydroxychloroquine works is like with cellular uptake. This just shows anything being taken up by a cell. And it's taken up, and the cell forms a little membrane around it, and it joins with other endosomes and fuses, and then it's usually nutritional, so it degrades because through a lysosome and releases nutritional particles. And that's the sort of the basic science underneath this. And in malaria, the trophozoite, the malaria trophozoite, which is in the blood, is very much affected by chloroquine. And it can't take up things from the environment, so it dies. So in areas where the malaria remains sensitive to chloroquine, there are zillions of people taking chloroquine once a week to prevent malaria. Okay, so somebody said, well, gee, if it prevents the uptake of any ligand at all, maybe it'll prevent the virus uptake, prevent this coated vesicle. It also changes the endosomal pH, and this fusion doesn't go on. It doesn't take place. So maybe we can just slow down the process, because the whole idea is that the virus starts to multiply in crazy numbers, and then goes to lungs in crazy numbers, and then there's a huge cellular response called cytokine storm, and you get your pneumonia. And if you could just think of it like a battle, where you want to pin down the enemy and then call in reinforcements. So what you want to do is you want to pin down the enemy in the nose, and then let the natural immune system make its antibodies and its T cells, its lymphocytes, and come to the rescue before you have this massive battle in the lungs. So keep the enemy pinned down. This was not a crazy idea. And the way hydroxychloroquine works, if you just want to illustrate it here, is this protein here, clathrin, this is the fusion protein, is inhibited by the chloroquine. So it was worth a look, okay? It was just worth a look, nothing more than that. So it seemed to work in vitro. In the dog, in vitro, the cells of the dog, but you need 6,700 nanograms per ml. While the human therapeutic concentration is less than 100 nanograms, then you start to see toxicity. So okay, so let's modify our theory a little bit and say, well, we're not going to cure it, we're not going to prevent it, we're just going to slow it down with ordinary doses of hydroxychloroquine or chloroquine. Reasonable idea, needing a reasonable look. Now, when I heard this, my first reaction is, oh, this is a piece of cake. Just look in Central America where half the country is taking hydroxychloroquine, see if their disease is milder. If it is, you've done your experiment. But everyone went crazy, and they had to do studies and studies and studies, and that's what they did. So ultimately, of course, the study showed, I mean, the hysteria was, on one side, the news media was saying, hydroxychloroquine, dangerous drugs. They prolong the QT on the electrocardiogram, and they're going to kill everybody. That was one side. And the other was huckster saying, it's the, using regular English words, saying, oh, it's the game changer and all this stuff like a huckster would do. So it was true mania in a reasonable scientific area. I just looked at it. I think the word is bemused. So then they did a study. Somebody did a study looking back. And this was published in the Lancet. And here's we get to the Gonif. And this is the title from the Lancet. Now, Lancet is a very good medical journal, very good medical journal. But it is their place in life to have the first article on everything. And usually, it's wonderful. You get the first good view of something. And it's very helpful. And in this case, they published an article, hydroxychloroquine, with or without a macrolide, that means like Zithromax, for treatment with COVID-19, a multinational registry analysis, meaning they're taking other people's data, which is always good because you're not creating data. And the person of greatest importance here is Sapan S. Desai. Okay, Sapan S. Desai. He's an American. Now, things my father taught me, things your father taught you are often come back as very important. Gonifs are born, not made. Okay? They don't become dishonest one morning. They are that way. Gonif principle number 2. Gonifs always leave a trail. They always leave a trail. Gonif number 3. No matter how attractive the deal, never do business with a Gonif. Now that means never. That's my father's name, Albert Slate. And that's what he would talk about. Now he was in a business, he was a lawyer, but also a businessman. He developed properties. Malls, you may have heard them, like Mall of New Hampshire, the Meadow Glen Mall, Liberty Tree Mall. He did business with 7 other men. They worked together for 40 years. Never had a written contract between them. So they were not Gonifs. Now, let's look at Sapan S. Desai. And this really should have been done by the Lancet when you get an article from somebody you don't know. Okay, and this was like a 2-hour job. You look at the website on WebMD. It says, Dr. Desai is the author of more than 200 publications in textbook in medicine. Now, he's a pretty good-looking young guy. Well, how old is he? He's 30. This time he was 38 at that time. Well, how do you get 200 publications in textbook in medicine? Even Charlie didn't get 200 publications in textbooks in physics. I mean, how many publications did you have, Charlie?
I have no idea, but it might be 100.
100. Well, you're way behind him because at age 38, he had 200 publications and in textbooks of medicine. So he's a very productive fellow. Now, he was also a neurosurgeon in practice at the same time. So you can imagine how he did this. Okay, so let's do a little math. These are like these Fermi things you taught us, Charlie. He completed training in 2014. This is Charlie's atomic bomb course. 200 textbooks and publications and textbooks and medicine, mostly after residency. That comes out to be 30 to 40 publications per year. That's making you academic people look kind of weak. Okay. While working as a vascular surgeon, while running his companies, Quartz Clinical and Surgisphere, which just started as a medical textbook company, I admit that. Okay. But you got to wonder. You're already wondering. So let's ask around a bit. Now, this isn't hard to ask around. A chief resident from Duke, that's where he trained, said you couldn't trust what he said. You would verify everything that he did and take everything he did with a grain of salt. That is hardly an endorsement. And then 13 people interviewed by the New York Times, this was a little harder, said there were broad concerns inside the surgery department at his hospital about Desai. Now you're finding stuff, but this is all innuendo and people are jealous. Who knows? So let's look on Amazon. There were fake 5-star reviews on Amazon from accounts impersonating actual physicians. This is never good. Okay, this is a Scientist magazine. His doctoral advisor at the University of Illinois said he told her he was enrolled at John Marshall Law School. The school has no record of him. His spokesman said the degree is not on his resume. So, really?
Enrolled doesn't mean a degree.
I'm sorry, the degree is not. I didn't mean his resume. It was not on there, on the records. That's what I meant. I'm sorry. So now look at his publications. Now he did a PhD paper. Now you can't. It's a strange topic, rodent inner ear, but you can't blame him for the topic. That's his lab guy and his mentor. He does. He's just a grad student. You know, he can't control that. But let's look about data manipulation. He shows some slides, and if you look here, this is part of the inner ear of the rodent, and these dots should be random. These are biological structures. If you look at what's in this green circle here, and this green circle here, these are the same. If you look at these two yellow circles, these are the same. See these two dots there? These aren't even rotated. They're the same. See these here? Same. These here? Same. That's impossible biologically to happen. Oh, maybe it did. So let's look at another slide. No, it's a tree squirrel. Where you guys? Tree squirrel, I don't know. See this here? These here? These are the same. These two are the same. Now look at this. All of these are the same. He didn't even rotate them. I mean, give me a break. They always leave a trail, you see. And the same here. So what you have is falsified data. Now, if you had the primary image, the metadata would show that it's an overlay. Well, I can't show it on here, but the metadata would show it's an overlay if somebody were to look at it. So his honesty has just gone down the tubes. So let's look at his Wikipedia page. He started out as a computer consulting service, just teaching high school students to access the internet. That sounds legit to me. And then he did some scientific illustrating publishing, probably legit. But now he starts to show that moss is not growing under his feet. He brings Apex testing, trying to help kids pass tests, which is okay. And then he gets into it. Quartz Clinical. Now, this is jargon, but cloud-based healthcare data analytics and machine learning. Quartz Clinical precision is always a good word, precision analysis for medical organizations who need to measure and increase performance with precision. You're not supposed to use the same word twice, the same paragraph, but that's okay. Okay, and then Quartz Clinical becomes merged with Surgisphere, which is hospital management consulting service. And what they said they did was they accessed hospital data systems to give them useful data. For example, an administrator wants to know the length of stay. They want to know cost per patient day. They want to know cost per patient day in pharmacy, cost per patient day in nursing. They want to know against diagnosis. They want to know lots of stuff. Now, there are lots of overlay programs. This wouldn't be unusual. There are lots of overlay programs for databases because you have a data field and you access the various data fields and you conglomerate them. You see the advertisements for Smartsheet on the television, but there's a million companies, even in the local area, that do overlay programs for your database and provide you the information that you need as a manager. And I was involved in a couple of them. I was involved in Meditech, and then with two overlay programs. One was Iatrix Medical Systems, which was a couple of engineers from Meditech. And they started, because Meditech's lousy at extracting data, and they had an overlay program, which I worked with them. And when I first worked with them, being suspicious that I am, I wanted to look them up. And I didn't actually, I heard their name, but I actually didn't, they're from Boxborough, Boxford or something. I didn't know how to spell it. So I just Googled them, IATRIX, Iatrix. And the computer said, this site is blocked, it contains pornographic material. I said, that's the kind of consultant I want. However, it was IATRICS. It was much more mundane. And what we did was we went into the database in Meditech, which is very clearly marked, and extracted things that the pulmonologist would want to see so I could get reports on the patients on ventilators very quickly on lots of details with tabs. It was wonderful, but it was a lot of work going down to each of these proprietary data fields, seeing what they said, and then deciding what you wanted. because Meditech names them for you. That is not, they come customize a lot of it. You can't just go to a hospital system and by artificial intelligence look at the data fields because they're named after kids and dogs, and you can't figure it out. And then I worked with Meditech with the lab in Cambridge. What's the big lab? What's the big medical lab? Wait a second.
Was it Bioran?
No, the one that does all the medical stuff, Quest. And we had wanted their data to go from Meditech to want the lab test to be ordered in Meditech, to be run at Quest, and be put back into the Meditech record. And we had to create all sorts of links. It was very confusing to create links to get their database to match our database, both going out and coming back in, so that when you generated an order, it created a mailbox for the results. Well, that was a big chore to figure out what Meditech had and what Quest had. And you don't do it with a very small company. You can't do it with artificial intelligence because you don't know what it says until you dope it out and see what it says. So now Surgisphere, they claim they analyzed medical data from 500 hospitals, automated to extract from the EMR and billing. Really? I don't think so. 500 hospitals. Databases save data all over the place in all different ways. They require the data fields to be linked. It's a laborious task. An employee at Surgisphere, a former employee, recalled only one hospital be signed up in 2019. And Dun & Bradstreet reported the revenue, I think it was for 2019, of $45,000. Well, if they 500 hospitals, $45,000, that's not very much per hospital. This is not wrapping up very well. They could have got a Dun & Bradstreet report. So let's go back to the paper in Lancet. Sampan S. Desai did not show the primary data to his co-authors and refused to provide the data to an independent reviewer of Lancet. And this is a common thing when people have proprietary data that the neutral reviewer just will keep it secret. This is not a problem. And the hospitals would have agreed anyway. He claimed that confidentiality agreements precluded his showing the primary data. Well, that was his undoing. And
They were probably under audit by the IRS.
That may be. For $45,000, I know he'll be paying $750 in taxes too. So the number of cases reported from Africa made up 25% of the total, and his African hospitals only represented 5% of the total hospitals. It didn't make sense. Nothing made any sense. So I think, this is my thinking, he was just trying to make Surgisphere seem like a major company and acquire hospital business by faking a medical article, because he's faked many before and he never got caught. So seemed like a reasonable approach to me if you're Gonif. Is Surgisphere still in business? Probably not. The website is gone, but don't worry, he'll be back. They always come back. And that's my Gonif story, and I'm sticking to it. So now,
Jerry, a quick question about tonic water or quinine water.
Yeah?
It's effective against malaria, right? It's sort of a palliative thing, or not?
Well, quinine was the basis. Quinine was the basis of homeopathic medicine, if you recall that. Homeopathy, which was very popular around 1900, and it was by Hahnemann. And it was a very popular concept because medicine itself was so ineffective. And what it was that like cure, if given in very minute doses, cures like. And quinine caused a little bit of a fever and shaking. And they gave it, and of course it made malaria, it prevented the malaria, and maybe cured a little bit. And so that was the basis for homeopathy, which was just as good as regular medicine, because regular medicine wasn't any good anyway. So they gave things in quote, homeopathic doses that were harmless, and they had the same results as medicine did, because it wasn't harmless, but it didn't do anything. So that was how quinine got started. But the amount you can get in quinine water is not enough to be preventive for malaria.
Okay, thanks. My dad used to tell me about quinine water.
Well, they have it. Now, so now we want to get back to go back to the real world.
Well,
rather
I was
than
going to add a story about getting 200 publications.
Yeah.
There are documented cases in physics where both the original article and the retraction are listed as publications.
Now that is good. That is good. Let me see if my ink is. No, it's not on. Let me see if I get my pen on again. Pen. I don't know why my pen isn't on. Pen. Let's go back to the. Oh, there it is. Okay. Let me go back to the beginning here. So now we're going to talk about science here rather than the fun part. And we're going to talk about the COVID update. And everybody knows about the COVID virus with its M spikes here that are going to get you. And here's the COVID M spikes here. These are the M spikes here, and these are the parts that are going to attach. And these were well worked out in the very early part of the year. And this is the bottom here, and this is the active domain here that attaches. This area here. And real science is this. And the way the COVID virus attaches to the cell, and this was worked out a long time ago because they looked at the old SARS and the new COVID-2 SARS. They're really the same how they do it. What you have is you have your M spike here, and the M spike comes up to the cell, and you have a serine protease. Now there are like 170 or whatever serine proteases in the body. They do various functions, but there's one on the cell membrane, and this serine protease acts at this point enzymatically to facilitate this attachment to your angiotensin II converting enzyme receptor. And that's a very important step in this virus attachment. M spike to angiotensin converting enzyme receptor using the endogenous serum protease. So again, showing here's viral entry. Here's it's going to attach to the cell, and then it's going to form this endosome here, this particle here, and get into the cell. And then when that happens, the cell is under new management. Now we're going to get a little bit more complicated, but not that much more complicated. Graphics can confuse you, but not as much as paragraphs do. So this shows here, it shows the virus coming here. Here's our serine protease here, and it's angiotensin receptor here, and it comes in the cell. We've got our endosome here, and it's going to uncoat, and it's going to release its genomic RNA and some RNA to make proteins here. It goes to the ribosome, and it makes two things. We'll just skip up to here. It makes the genomic protein here, and it makes some other proteins here that are going to go into the endoplasmic reticulum and then to the Golgi apparatus, which are intracellular structures of some interest to me. And ultimately, the protein coat is going to be formed here, and it's going to take the RNA there, and it's going to make a virus. It's going to be released into the body to infect more cells. So there's a whole bunch of spots now. There's a whole bunch of steps where you say, well, can I interfere with those steps? We talked about one that didn't work with hydroxychloroquine. It didn't work, but it was a thought. So now on this very cluttered slide, which looks like it'll make you crazy, but there's only a couple of points to it, they cleverly colored this to relate to this. So we can look at blockade of entry. Are there points where we can blockade the entry? Well, maybe we can make a spike antibody that'll lock up this spike and prevent it from getting on this receptor. That's pretty good. Or you can interfere with this cleavage in here. You can interfere with these various steps. Another place that has been interfered with here is with the protein production. a blockade of replication, where you inhibit a replicase. This is what you do for the DNA virus of HIV. You block this replicase. Or you can fool around in here with the RNA production here. You have to fool the reader. You have to fool the production or the reader, which is a little bit difficult. This is why remdesivir has trouble, because it doesn't really fool. It interferes. It gets into the RNA, but then it doesn't fool the proofreader. And then, or maybe we can interfere with some of this stuff over here, and maybe we can interfere with the cytokine storm. Just don't make so many cytokines, and you'll be a little bit better. So those are the kind of the different areas that people look at. Now, on the news, they talk about 3 vaccines being produced, which always mystifies me. They make it their business to be ignorant. The London School of Hygiene and Tropical Medicine details 218 vaccines in development. So when people are yelling and screaming, oh, well, there's not going to be enough vaccine. Oh, Moderna can only make 300 billion of them. Well, you got 218 companies all vying for this stuff, and some are going to be some are going to be successful, and they're hoping that the immunity doesn't last too long, so they'll be inoculating you every year for $20, and it'll be a good business. So there are so many people trying to produce this stuff. When people say, "Oh, it won't be available," that's sort of crazy. Now there are only so many. So we've talked about the methods, but there are only so many ways you can skin a cat. And put this more into English than into sort of science. You can use an inactivated or killed virus vaccine. Now, you can't really kill a virus because it's not alive. It takes over the cellular machinery, but that's the expression that we all use. And you kill it in one way. You put it in formalin, which is a mixture of formaldehyde and water, or you put, who knows, you heat it up. You make it killed, but it has enough viral structure. It has the M spike, so it does create immunity. Or you could modify a coronavirus, make it attenuated and provoke an immune response. Or maybe you can make an unrelated DNA vaccine to go into your own cell genome and tell it to make the spike protein, which will be released into the serum, and you'll make antibodies against it because it's a foreign substance. Or maybe you'll somehow squeeze in messenger RNA itself into the cytoplasm. Messenger RNA doesn't last all that long. So if you can convince it to make some M spikes, and then they'll be released into the environment, and you'll become immune to it. You'll create antibodies. Or you can use some viral vectors to get in some messenger RNA or something. Or you can do recombinant technology, where you ask a bacteria or a yeast to make a particle for you, and then you inject it, an M spike when I say a particle. And there are novel vaccine molecules that can bind to a site, meaning to bind to the M spike, so it can't bind to the cell. And these are the approaches that people are taking. Now, here's where the real science is. This is really good stuff here. At least if you're if you're interested in this stuff. Over here is the M protein, and down here is the base. This is just the base here. It's just structural that holds it up. And here's the receptor binding motif right here. This is the action. This is what binds to the angiotensin converting enzyme receptor 2. And these specific helices, H1 and H2, fit right in here. And this is where the binding action is. This is where all the focus is scientifically on what can you interfere with this interface. So again, the showing here, can we interfere with this interface right here? So this would be one of the things we can do. Now, going a little deeper, you can make monoclonal antibodies. You ask a rabbit or some animal to make them for you, or bacteria to make them for you, and they're humanized. They're used, so they're not recognized as foreign, and they bind to this M spike here, and simply inactivate the virus. And the same approach comes from convalescent plasma antibody. You take somebody who's already had the disease, and you just take their plasma if they have good titers, and you hope that some of them are neutralizing antibodies, which is of course the issue, and that these neutralizing antibodies bind to these M spikes, and they prevent binding. But they provide only a short-term immune response, because they only last a couple of months. But there's still a response to the virus by the host. So now you gain immunity and you're not particularly ill. Good thought. Good thought. So now, we want the host to make antibodies against the M spike. One company that you hear all the time in the news is AstraZeneca. And they're using a benign virus, which we'll go into, with an added gene to make an M spike protein. They gave it to monkeys. They reduced the viral load and prevented pneumonia in 6 monkeys. The control monkeys all got pneumonia. But it didn't prevent illness. They had nasal symptoms. They had virus in the nose. So they prevented illness, which is a good thing. But they didn't prevent the disease. And they didn't prevent transmissible disease. But they went on with the human trials because it's a huge step forward if you can convert deathly COVID to a common cold. Serum Institute of India, which just recently got in the news, but it's a big outfit. They've been around forever. They have done the old tried and true method. They killed the inactivated the virus, gave the dead virus. This is like Peter and I were polio pioneers, right? Aren't you, Peter?
See, I didn't understand the question, Jerry.
Aren't you a polio pioneer? Did you get the first? So we got the first kill vaccine from Salk. And so this technique is tried and true because we're both still here. It prevented illness in the rhesus monkey.
I was in that too. Unfortunately, I was in the control group. So
how'd that work out?
Well, it made me fearful of needles for the rest of my life.
But you can still stand up and take a bow, so it did work.
True. And I did get over it eventually, but I had to work at getting over the needle phobia.
Yeah, but still you'd be proud you were part of the original study for this.
Sure. Yep.
This is a big step.
Just the flip side of that is that I actually got the vaccine, and I learned that I was allergic to penicillin because of it.
Okay. So this is like the big step since Edward Jenner. So they gave the killed virus, and it prevented all disease in rhesus monkeys, and no virus was found in the immunized monkey. Now, killed vaccines usually do not give lifelong immunity, which is why we went to the Sabin vaccine. We did, but still, it's a huge step if you can, prevent it for a year. And then we have the Russian vaccine, and this is Russia, so nothing is true and anything is possible. So let's look at Moderna because they get a lot of press. They're a Cambridge outfit, local guys. They're Israeli, or the chief guy is Israeli. So what they do is they take an M spike messenger RNA, which they made because and then they make their own proprietary envelope. Here's the envelope here. This is not biological. They've made this, and it fuses with the cell and creates and delivers the messenger RNA here. Then the cell machinery, the ribosome meaning, makes the M spike protein. Then the M spike protein goes, it's manufactured here, and then it's released into the circulation, and it's a foreign substance, and you make antibodies to it. So it's pretty good. 2 injections, 1 month apart, 30,000 people in the study. And I just marked this down, but it doesn't mean a lot. July 27th was the first day, but they didn't do 30,000 people the first day. So it doesn't really have much meaning. Because what they're really looking for is not 30,000 people. They're looking in the control group of 15,000 to see, 300 or 400 cases of COVID. Because once the control group has enough cases of COVID, then they can compare that to the treated group. Unless it's so obvious, if nobody in the control group gets it and people start getting it in the control, and nobody in the treated group gets it, and people in the control group start to get it, it could end early, but they estimate they need about 400 or so cases of COVID in the control group to make a comparison. So,
Jerry, that's true for all the current trials, correct?
Well, it's mathematics. Yes, it's math. It's not numbers. Correct. It's not the drug. It's the math to make a valid statistical point. And so you just can't so the animal trials are important, but mice lie and monkeys exaggerate. So you can't just use them to get to jump into human studies. So the human studies, there's 3 things that throw off the human studies: details, and details. Those are the 3 factors we look at. So you look at the Indian one, and there's one from China. So you use your dead virus, as it were, non-replicating, old technology, and it works. Sinovac, the Chinese company, is doing it in Brazil because they have a lot of people with COVID. Makes sense? Now, I tried to get the data on this. I really tried. So now, it produces neutralizing antibodies, which is good. Neutralizing antibodies have to be there. What is the duration of immunity? Nobody knows yet. And it requires a lot of virus culturing. This is why the flu shots run out, where there's enough influenza vaccine. It's hard to culture tons and tons of virus. That's a biological process. It's hard to make it uniform. You have some, you just have technical problems making good batches. It's not as easy as it looks. Now, using a vaccine for DNA genetic coding, where you take some DNA for the spike and you put it and you inject it and create a vaccine, that's never been done in humans, and it can be hard to get into the human genome. That too is not as easy as it looks. There are some animal vaccines. It was done in monkeys, but again, it protected them from pneumonia, but not nasal infection, which means they could transmit disease. A very important point. And Inovio is doing it. They begin their phase 1, phase 2 in April, and they have neutralizing antibodies. Sanofi GlaxoSmithKline is trying a DNA vaccine. Their phase 1, excuse me, began on September 1st. So I don't know where that's headed because it's only September 30th now. And the RNA vaccines, which are more in the news, you harvest some COVID RNA, the spike protein, and you find yourself some vector and put it in the cell. So old technology. Now, the virus, Russia has done one. Now, Russia is using, now, I don't know what they're doing. I only know what they say they're doing. What they're saying they're doing is they're giving 2 injections. They're giving adenovirus 26, which is a simian virus, which is non-replicating in humans. And then, I guess a month later, they're giving the same messenger RNA in adenovirus 5, which is a human adenovirus. The problem with the adenovirus 5 is that 60% of people have antibodies against it, and that part probably will not be an adjuvant. And it doesn't work out just to give massive doses because people get too much of an inflammatory response because they're ready against that adenovirus 5. So that doesn't sound like it's going to be quite what they hope, unless the first shot portion, the adenovirus 26, works out. And Merck used this adenovirus 5 for a viral product, and it increased HIV susceptibility. Whether that would be relevant or not is unclear, but that is sort of hanging out there using adenovirus 5 viruses. But with Russia, who knows what's going on? So what's going on in the Western world? We're using adenovirus 26, non-replicating in humans, monkey virus. There are no antibodies in Europe, and I think it would be fair to say that there aren't any in the United States. 9% of the people in Gambia, where the monkeys are, 9% of people have these asymptomatic infections, or at least they have probably because they have antibodies to the virus. So the Oxford University, the Jenner Institute there, Jenner is the guy who did smallpox, they gave 6 monkeys the nasal infection. And again, here's the deal, which I alluded to. None of them got pneumonia, but 3 of the 6 monkeys had nasal infection with virus that was sheddable. So they've gone on to their human trial, and one patient, which I'll show you in the next slide, developed transverse myelitis. Johnson & Johnson are doing the same approach, as I said, adenovirus. And Russia has the adenovirus 26 a vector by adenovirus 5. You figure it out. So let's look at what happened at Oxford. I talked to a guy I know at Oxford, and they're very concerned about this. They're keeping up a brave face, but they're very concerned about this. The patient Now, show of hands, did anybody here ever hear of transverse myelitis? Nobody ever heard of it. Okay, there's a good reason you never heard of it. It's a rare disease. Now, this is an MRI, which I'm sure you're used to looking at, but I'll explain it anyway. This is an MRI of the spinal cord. Here are the vertebrae in the front. Then there's the spinal cord here, and then the vertebral processes here. So this is really a tube that you're cutting down on. And the spinal cord should look like this gray right here. Here you have signal right here, where it's abnormal, it's inflamed. You see the signal. So then you're going to take a cut through this signal here. I'll use my pen here. And then you're going to look at this cut over here, and this is the inflammation, this white stuff. It should all be dark, like it is in the periphery. And the person develops some degree of paralysis. Now, this was a 38 or 28, I think 38, 38-year-old woman. Now, transverse myelitis, it's 1/3, 1/3, 1/3. 1/3 gets better, 1/3 has some residual, and 1/3 has severe impairment after this transverse myelitis. This woman was discharged from the hospital, so she's probably in the top 2/3, the one either is going to get totally better or be left with some mild impairment. So transverse myelitis, the incidence is 1.4 per million, which is 1/10⁶. Charlie told me to do that. Associated conditions are multiple sclerosis, cancers, autoimmune disease, and a few other diseases. But it's 1 to 4 per million. Then there are infections, Lyme, mycoplasma, herpes, hepatitis B, influenza, some fungi, some parasites. And the vaccinations, which seem to be similar to the infections that cause of the same agent. So 1 to 4 per million. Now, let's look at but let's drill down a little bit. Since 1980, there have been 39 cases occurring within the first few months after immunization. That's how many have been reported. There may have been more because I'm only talking about how many reported, not how many happened. But the sum numbers are so low, it's hard to make a clear association. But when you look at the overall incidence, it does rise a little bit after immunization. So it's a very rare side effect of immunization. But 39 cases reported since 1980. Now, we give, now we're doing another Enrico Fermi problem. When all other known, there's the Oxford restart. They've restarted in England. When all other known causes of transverse myelitis are eliminated, let's just say this is about 1 in a million, because 3 up at the 4 per million, but I've eliminated all the other causes. They don't have Lyme disease, and they don't have this, they don't have that. They didn't get another shot. Now, 10,000 people, 10 to the 4th, received the Oxford vaccine. So this chance of this being a random event is about 1 in 100. Now they say, let me see what's on the next slide. Yeah, they say there's no proof that it was due to the vaccine. Well, they eliminated all the other causes, but they say there's no proof. Well, that is true. If you don't believe in the Arabic numeral system, there's no proof. Now, it was restarted in England. The CDC has not restarted in the United States. So, I thought I had another slide there. I guess I don't. So, it's hard to know why they're doing that, but they didn't start it, but they haven't said why they haven't restarted it. Now, I was going to talk about some man-made vectors. We talked about Moderna, and the Moderna makes the messenger RNA to make that. But BioNTech and Pfizer have joined to make a nanoparticle. The cell makes the M spike. Now they have 2 products, 162B1 and B2. The B1 is just this part here, the right here, the receptor, this binding motif right here. And the B2 is the entire shebang. And they've gone forward with the B2 because the full-length spike antigen had fewer side effects, which they didn't tell you what it was, than the B1. So this is now in phase 2 or 3. You never see this in the news, but they're doing this one. Now, I have some good news for seniors from Moderna and senioritas, I guess. They're neutralizing antibodies. This is young people. These are 18 to 55. These are the antibodies that this is what happens in COVID. And this is what happens when you get their vaccine. So they make more antibodies. That's pretty good. And there's convalescent antibodies. And then you look at the middle-aged people, 50, 60, 70, is the same. And you look at old people, we're all the same. So that's pretty good. We don't need, so it's not age-related, so everybody qualifies. And again, they looked at their T cell responses in the young, the middle, and the age. And sure enough, the T cell responses between day 1 and 43, very young, middle age, old, all the same. So age is not a consideration in the virus, in the thing. But again, the details, the Moderna vaccine requires storage at -4 degrees, or -20 Celsius, for anybody who understands the metric system. Pfizer, -24, as far as our international audience. Pfizer, -94, which is -70 Fahrenheit. And dry ice, for your point of reference, is -109, or 78 degrees. So the term that's coming out now is called the cold chain. Can they really make it, transport it, and deliver it with an unbroken cold chain? The Pfizer one, I think I heard, will last 48 hours. Maybe it's Moderna, I don't remember. Will last 48 hours in a regular refrigerator. So if it's received on site, it can like the town of Lexington or whatever, they can just keep it in a refrigerator overnight. That won't be an issue. But can you really ship it from wherever by UPS and rely on everybody not to break the cold chain? Well, we can't even make face masks, so I don't know if we can. If we can do that.
Jerry,
what happened?
you say that
Stored that.
Go
ahead, Mike.
You talk about how long it can last, with a cold chain, and you're making it seem like an on-off switch.
No, this is that it's broken. The cold chain must be continuous.
No, I understand, but you say, well, it can last for so-and-so time.
48 hours. I don't know if it can be refrozen.
What does that really mean? Because there's a certain amount of degradation during that 48 hours, isn't it?
There has to be, but I guess there's enough concentration. I don't know if you can refreeze it. They did not. I didn't think of it, first of all, and second of all, I don't know if you can refreeze it once you've frozen it. I don't know.
Jerry, what happens if it's kept at a colder temperature than what you indicate? For example, it's all stored in dry ice.
It will be.
But
it'll be colder.
That will bring the temperature down to 109 or -78.
That's all right. It's just less. It's just not shaking around as much.
Okay,
right.
so if you physically don't interrupt the virus or the drug, and you keep it at a very, very low temperature, it can be transported and maintained for a reasonable long period of time.
Oh yeah, for long periods of time, sure, because there's very little molecular movement, that kind of thing. But it's a challenge moving things, and people do funny things, and they take the weekend off, and I don't know. When you see all the discombobulation that goes on, maybe Amazon can do it. I don't know.
Does movement mean dropping it from a truck, or movement means just shaking hands with somebody type of movement?
No, it means moving it on a truck. But I don't. It's just a lot of logistical problems trying to get it all around the world.
Understood.
Just a lot of logistical problems. But the term I wanted everybody to learn was cold chain. Recombinant technology inserts the gene of a molecule into a cell, which produces more of that molecule. That's what it is. And you can use bacteria, yeast. You can use tobacco leaves, and lots of cells. So you can take, say, this is the M spike protein. This is from the genome virus, the genome, and you put that into a yeast. I'm just making this up. And you put it into a yeast, it'll make you your M spikes. Now, Novavax, they have a proprietary technology. That means you can't find out what it is to make a full-length M spike nanoparticles through some recombinant process, and antibodies against that are actively produced, and lymphocyte activity is actively produced. They've given it to non-human primates, but I read the preprint article, and it just didn't say how much protection it provided. I don't know why they didn't say it, but they just didn't say it. It's kind of mystified me, but I was wondering if they felt they shouldn't say it because if you say something, they can accuse you of stock manipulation. Either way, if you say it worked well or it didn't work well, then they'll look and see if you bought or sold the stock. And whereas if you say nothing, you can't be accused of fooling the public. And then at University of California in San Francisco, they've created a novel molecule which they say is simple to produce. I'll take their word for it, which binds to the end of the M spike. And first, just look to the right here. This is the novel binding here to the top of the M spike where it tries to attach to the human cell. And this is just a top-down view. It actually binds to 3 spots of the binding domain. So they think that may provide some transient blocking of disease to make the disease milder. So now there's some lingering questions, which long-term safety. You can't save 5 miles on a 3-mile trip. You can't know the long-term safety until you've given it long-term. Short-term efficacy, we should know that fairly soon because they're moving along in the trials. Question, because the minor coronaviruses we don't maintain long-term immunity to those viruses. And now here's the problem about nasal infection and herd immunity. If you give a vaccine that if you get the COVID virus, you're either going to have no symptoms or a minor cold. You can still give it to others. Well, if you get the virus and it can only cause a minor cold, and I've had the vaccine as well and only get a minor cold, big deal. Life goes on. I'll go to the restaurant. I'll take my chances like I do with every other cold. But you're going to have a whole bunch of people, maybe, who are going to say, well, I don't get immunized and I don't believe it and I'm not going to do it. So now my question is, if you were working and you're going to work every day and you've had the vaccine and you get a little cold, are you going to go get a test for the COVID virus and self-quarantine for 3 weeks so that you can prevent people who've decided they're not going to get the vaccine you're going to protect them while you stay out of work for 3 weeks? Americans are cowboys. I don't think we're going to do that. What do you people think? Do you think people will stay home? So we won't have herd immunity until after a whole bunch of people get it because they're
already not staying home.
Yeah.
Right, right. So it's not going to work out. Although
Gary, it isn't going to be framed that way.
That's what I was going to say. What do you think? Go ahead, Mike, because that's what I was going to say.
I said it's not going to be framed the way you did. You were very specific about saying that are you going to stay home so that you don't infect people who refuse to get the vaccine? That isn't the way it's going to be framed.
How would you?
I have a feeling
it's going so that you don't infect others.
I have a feeling it's going to work the same way any legal waiver operates. If you choose not to get it and it's available, you have to sign a waiver.
Well, look at it from an employer standpoint. Look at it from an employer standpoint. The employer has let's say a big employer and has experience-rated insurance. Are they going to let you come to work unimmunized and get really sick and be on their health insurance? They're going to say it'd be like the flu now. You can't work in a hospital without the flu shot. They're no longer interested in a song and a dance, so you can't do this and you can't do that. They're just no longer interested. Say, you don't have the flu shot, just stay home and you won't be paid. That they've taken a hardball approach to it. And I think that many employers will take a hardball approach. They just will say, you can't work here and do that. And
Jerry, like many other things, it becomes regressive in terms of its efficacy. People who have to work the most are exposed the most and have the least control over their life. And, the guy flipping burgers at McDonald's is going to show up and flip burgers, even though he has open sores.
Yes, but will he have had the immunization?
Doesn't make any difference. He's showing up because he got to feed his
Right.
family.
Right,
Or
he'll
she.
show up, but if he's had the immunization, he's not going to get too sick, and he may transmit it to others, as may you, but he isn't going to get too sick. I'm talking about the health insurance.
You go down the rat hole to if he has health insurance, blah blah blah. There's a whole.
It's going to be Shaw's going to be free. Shaw's going to be free.
And you
Oh,
gotta,
the insurance. Oh, well, they
have.
have insurance at McDonald's. They have insurance at McDonald's. Okay,
You know,
but anyway, it's going to
whatever.
it's going to be a Donnybrook, and I don't know how it's going to come out. Now, I wanted to show you just an interesting image. This is scanning EM. This is here on the left is early COVID infection. This is a lower power, EM, which is really high power. Here's the COVID viruses scattered around here. And then this is one is 4 days later at a much higher res, but there's a lot more COVID virus sitting on the respiratory epithelium. It's multiplied. So it's just interesting image. And that's it.
Jerry, thank you. I just one question, which is a biology question. Very good. A lot of people take, I think it's called an ACE inhibitor for things like blood pressure and other things like that. Is that the same as the ACE that you were talking about in your presentation?
Yeah. Angiotensin converting enzyme receptors are all over the place. It's not clear. They're on the blood vessels, and that causes them to when angiotensin gets on them, cause the blood vessels to constrict. So when you take a blocking agent that blocks angiotensin production, like lisinopril, you have less angiotensin around to stimulate those receptors. It doesn't seem to have any other adverse effects. Then there are agents
so how does
that block.
this interact with the virus that's working with the ACE receptor as well? Will there be an interaction that is problematic?
No,
Is that a question?
no. Let me go forward. That just prevents you from making angiotensin itself, angiotensin 1, angiotensinogen to angiotensin 1 to angiotensin 2. It doesn't like lisinopril or captopril, doesn't touch the receptor itself. The receptor is still sitting there. The question was with that, whether you would upregulate your receptors when you downregulate, which does happen in other situations, when you downregulate the agonist, the angiotensin, do you upregulate the numbers of receptors? And the answer is no, you do not. So it has no influence either way, and that's been looked at carefully. And then you have the angiotensin receptor blockers like COZAR and things like that. And they block the receptors, but you don't upregulate your receptors, and you don't block the virus meaningfully to getting into cells. So neither of those groups has any effect on disease incidence or severity. It was an interesting thought. It was worthwhile looking into for sure, but it was not didn't turn out to be anything.
So Jerry, on you're pointing out the M spike on the virus, and there's a lot of spikes on the virus. So is the M spike a particular one of those spikes?
It's all of them. It's all of them, Charlie.
Well, then I don't understand how you say, oh, I block one. You got to block them all.
You do.
You? Yeah.
You do. Most of them. Yeah.
So any vaccine has. I mean, how many spikes are there? I mean,
have you ever had polio? Have you ever had polio?
I may have. It was unclear.
Well,
I was a very sick puppy, but I didn't get any
paralysis. Well, the rest of us never did. So who got the vaccines? So the answer is we make enough antibody. We just do.
Yeah.
And antibodies are very small as compared to a virus. They don't show, you can't show it on a graphic. Antibodies are very small, and you can give zillions of them. And we don't get mumps a second time, and we don't get measles a second time. And you can take a hepatitis shot, and you don't get hepatitis. You can block viruses with antibodies. You tag them. The antibody doesn't just bind to them; it tags them for uptake by the macrophages. So they're doomed once they get tagged. They're marked as doomed. So they don't just hang around, floating around, waiting to cause another infection. They get eaten up fairly quickly, which is why vaccines prevent infection in us.
Mm-hmm.
It's hard to display those kinetics on a slide, but the virus is massively overwhelmed by the existing numbers of antibodies.
Thank you.
Jerry, can I ask a question? This is Dick. I've been following this COVID, fairly much since it started, but not from a medical standpoint per se. And my question has to do, have you seen papers about the transmissibility of the SARS-CoV-2 in terms of, in particular, in terms of its transmissibility from surfaces? In other words, once the virus gets onto surfaces, how transmissible is it in terms of its, particularly in terms of its causing disease? In other words, whether the COVID.
Well, the issue is starts with how long does the COVID virus last on the surface, not how long can you find the RNA. You can find the RNA.
That's not really what I'm asking.
Well, I'm getting to it.
Because you can have the genetic material there, and you can say that's the COVID virus, but that's not going to be
correct. That's correct. That's what I'm getting at. So you can't just swab a surface and look for messenger RNA and look for the COVID virus particles, what you're looking for, any component of it, and say, well, that's the COVID virus. You have to swab it, and then you have to give it to an animal and see if they can get infected. And it's pretty tough to get enough COVID virus. Nothing has an infectivity of 1, and it's pretty tough to get it from a surface into the nose to infect. It's pretty tough to do that. People like the idea of surfaces being clean, but the data doesn't support all this crazy cleaning. It supports the aerosol distribution. And if I could say a word on that, when they say we're just learning, and even Fauci said this, well, Fauci's no pulmonologist, that's for sure. Aerosols were very well studied during the tuberculosis era. And the transmissibility of tuberculosis, this is old information. And looking at particle size and what particle size you need for infectivity, for carrying and for infectivity. And there's 2 parts to any, create an aerosol. Well, a sol is a suspension of particles in a liquid or a gas. An aerosol is a suspension of particles in a liquid. I mean a gas, a suspension of particles in a gas, aerosol. So when you have a nebulizer, say, or an atomizer, make it simple, like a perfume bottle. When you have a nebulizer, a perfume bottle, you create an aerosol of many different size particles, polydispersed sizes. And the sizes that are 5 microns or larger, which is most of them, fly a little bit and have trouble going more than 6 feet. This is true. They just have trouble because they just don't hang up. They're aerodynamically dense. Then the particles less than 5 microns by Brownian motion, they get kicked along and they stay up in the air for a long period of time, hours and hours, and because they're aerodynamically non-dense. Well, if someone walks into a room with the COVID virus, coughs once, and walks out, then you're going to have the distribution of particles. You're going to have a small number of aerosols, a very small component of aerosols, and a very large component, of small particles, and a very large component of large particles. If I can just get my paint program up here. Am I sharing my screen? No.
No.
Let me share my screen for a second here. Okay.
Sorry, I thought you were done.
I am. I thought so too, but I guess I'm not. Let's see. Let me get a brush here. Okay. I get a better color. Now, if we look at, look at particle distribution. This is the number of particles, and this is the size. You'll see just a bell-shaped curve. And this will be the aerosolized particles. These are small, and these are the large. So these particles are going to fall down very quickly if they walk in the room, and these small particles will remain. But if that person stays in the room, coughing, singing, whatever, the numbers of large particles stays about the same because they keep on falling. Let's say they stay roughly the same. But the number of the small particles starts to rise because they're staying up in the air. So later on, you'll have a distribution of size more like that. Does everybody see that? Yeah. Is that clear the way I'm drawing it? Yes. So if you go into say a long thing like a wedding where people are there for 3 hours, well, you're going to have a lot of aerosolized particles drifting around with if there's a COVID person there, with COVID virus. And they're going to build up over time if the ventilation system doesn't suck them out, which usually they won't.
Which
So
of these is the COVID? Would it be the small ones?
These are particles. These are particles. These are just particles. If there's a COVID, they'll all contain some COVID virus. But these COVID virus particles will fall down, and these COVID virus particles will remain suspended.
So you're saying if they walk in and they give a single cough, they give out both the small and the large particles. The large particles drip down. The small particles are diversify, diffuse throughout, and become less effective. But if they stay there, they're adding to both of these. So there'll be more small particles that'll hang around longer, and the more large particles that will eventually drop down.
Well, they drop down fairly quickly, so they don't increase very much.
But the longer they stay, the more of these COVID-holding particles will be introduced into the air.
Right, but the small particle, the aerodynamically non-dense, the tiny particles, less than 5 microns.
Yeah.
Those are the ones that are going to build up. So when you see these, like there was that episode in Maine where they had a wedding indoors.
Yeah.
And they were there for X number of hours, I don't know how many, and then all sorts of people got the COVID virus. Well, that's inevitable because of that. So when they say, it's like my joke, I may have told this before, I think I did. The joke is that an internist, a virologist, and an epidemiologist walk into this bar. That's the joke. So the
medical joke, Jerry. Not everybody gets it.
Right. They wouldn't dream of doing that, because the longer the people are there, the more the aerosol, the more the 5 micron particles are up there, and the more infectivity of the atmosphere. Which is why
the point you're making, Jerry, is that it doesn't make any difference if the aerosol is large or small. You still have the propensity to get the virus. The large ones just keep getting refreshed by repeated.
That's a good term. I'm stealing that term. Refresh. That's exactly it. Now,
they get refreshed by continuous coughing and whatever, and the small ones just keep magnifying.
Exactly. Now, if you're outside, the dynamic is completely different, because the wind just dilutes everything so rapidly that you're always at baseline. It just get blown away. So the people who are outside really are at very low risk. It doesn't compare to inside.
Jerry, what about the ion filters, the air filters that some people have in the homes? I'm not sure if they have, bigger ones, for larger companies.
Well,
would that help?
Well, small particles, what you're dealing with is small particles get an electrostatic charge. And then you have a filter that has the opposite electrostatic charge, and they will stick to that filter. So you will filter, yes, you will filter out all particles with an ionizing filter. How efficient it is another question. And
that's the same as the N95 masks. The middle layer of the N95 mask is electrostatically charged, and it also assists in trapping the virus as it passes through the material.
Okay, so electromagnetism works. Once again, the magic of science.
And that's why they argue why you can't reuse them a lot, because the middle layer, the charge, gets dissipated over time, and there's questions about how effective the mask is once it's been re-sterilized, if you will.
Does it dissipate, or does it just get bound?
I don't know the right answer, Jerry. I just know that there's questions about efficacy after they've been renewed.
Because I know in the filters, the big filters they use for asthmatics, you have to either clean or change the filter.
Right. And
the only thing I know is those N95 filters are very tiring to breathe through, especially for nurses, But what I'm getting at, if we promote, if ion filters were promoted, so you could have large gatherings and then change the filters, when you're supposed to. I mean, to me,
it's tough to tough to have that kind of efficiency when you're looking at filtering out dust for the asthmatic. You don't need that kind of efficiency. Whether you can that could be a tough challenge in any large space.
So one of the morals of this, it seems, is related to restaurants. Now, we went to Legal Seafood on Friday. We sat outside. It was freezing cold, but we sat outside. We were maybe 8-10 feet away from the next table. But place
that's very safe. That's very safe.
Yes, but the restaurant was open for people sitting inside. And
Well, they must have had the immunization.
Yeah. It wasn't easy to see through the windows except for the people that were right at the edge, but they seem to be fairly close. And from what you're saying about the small particles, if there are people in there eating dinner for 2 hours, one would think that over time, the whole inside of the restaurant was going to get filled with particles.
I think the academic expression is you can't teach stupid.